Welcome to this explainer. I am so thrilled to have you here today. We're going to dive right into one of the absolute most fascinating biological journeys imaginable, the step-by-step process of human reproduction. Whether you're studying biology, maybe planning for the future, or honestly just curious about how we literally all got here, this explainer is designed specifically for you. We're going to break down the whole science of conception, taking it from microscopic cells all the way up to modern medical miracles.
Okay, let's dive into this. Here's our roadmap for today. We'll start with the building blocks, map out the window of fertility, witness the fertilization events, track early embryonic development, and finally wrap up with assisted reproductive technology.
All right, Section 1, the building blocks, Gametogenesis. Because, before conception can even happen, the body actually has to prepare the essential biological components.
But hey, quick question before we look at those cells. Do you actually know when a pregnancy can happen? You might think the answer is just common knowledge, but the truth is, human reproduction is incredibly misunderstood, especially among young adults.
Now, what's really interesting about this is the sheer scale of the misconception. This data comes from a demographic and health survey focusing on young adults aged 15 to 24. Take a look at that right after period row. Nearly 48% of women and over 50% of men incorrectly believe that the fertile window is right after a menstrual period ends. No way, right? Only around 30% correctly identified that it actually happens halfway between periods. And honestly, this lack of basic biological understanding is exactly why we need to lay out the facts.
So to really get that fertile window, we first have to understand gametogenesis, basically the creation of sex cells. And there is a stark, totally fascinating biological difference here. On the male side, spermatogenesis occurs in the testes. It kicks off at puberty and is just a continuous, massive factory line producing millions of sperm every single day. But on the female side, oogeogenesis is a completely different ballgame. It happens in the ovaries, but the process actually starts before the female is even born. Diploid cells mature into primary oocytes and just wait there, suspended, until puberty triggers menarche. It eventually stops at menopause, and the whole thing is so highly regulated by hormones that typically only one single functional ovum or egg cell is produced per cycle.
Which brings us to Section 2, the window of fertility. Let's really get into the details here.
Because oogenesis only produces that one mature ovum per cycle, the timing of human reproduction is incredibly, almost unbelievably specific. Let's break down the actual mechanics of the menstrual cycle, paying special attention to some vital NCERT key terms you'll definitely want to remember. It starts with menstruation. This clears out the unfertilized ovum and sheds the old uterine lining. Following this is the follicular phase, which you'll also hear called the proliferative phase. During this time, driven by gonadotropins like follicle-stimulating hormone, or FSH, structures in the ovaries called follicles start to develop. Ultimately, just one of these becomes a fully mature Graafian follicle containing the ovum. Then, we hit the critical moment, ovulation. This is triggered by a massive, sudden spike in luteinizing hormone, famously known as the LH surge, which forces the release of that mature ovum. Finally, the luteal phase, also known as the secretory phase, takes over. The leftovers of that Graafian follicle transform into the corpus luteum. This little structure is a powerhouse, secreting copious amounts of progesterone to thicken up the endometrial lining just in case a pregnancy occurs. So, putting it all together, when is that true window of fertility? Well, since sperm can survive for a few days in the reproductive tract, but the ovum only lives for a very short time, the chances of pregnancy actually peak from about 5 days before up until just 1 to 2 days after ovulation. And ovulation, as we saw earlier, typically occurs right at that halfway point of the cycle.
All right, let's move on to Section 3, the fertilization event. If intercourse does happen during that specific window, we shift to the actual moment of conception. And it is nothing short of an epic microscopic journey. And this brilliantly illustrates the incredible odds of fertilization. Think about this. Out of approximately 200 million spermatozoa in a typical ejaculation, only about 200, yes, just 200, actually reach the ampulla. That's the specific curved section of the fallopian tube where fertilization goes down. The sperm are swimming at a speed of 1 to 3 millimeters per minute, and they are quite literally being chemically guided. The cumulus cells surrounding the oocyte secrete progesterone, which binds to receptors on the sperm, sending them into hyperdrive and guiding them directly to the egg. Once they arrive, it's like breaking into a vault. The sperm uses enzymes in its acrosomal head to burrow through the egg's tough outer protein shell, the zona pellucida. The plasma membranes fuse, the genetic material merges, and boom, a brand new single-cell organism is formed, the zygote. Now for Section 4, Early Embryonic Development.
That single-celled zygote doesn't stay single for very long. But as the cells begin to multiply, we actually need to clear up a super confusing medical concept. If you've ever known someone who is pregnant, you've probably heard doctors talk about how many weeks along they are. Well, doctors use gestational age, which is measured all the way back from the start of the last menstrual period, or LMP. Historically, they just use this because a period is an easily observable event. However, embryonic age is the actual biological age of the embryo from the exact moment of fertilization. Because ovulation happens about 2 weeks after a period starts, embryonic age is basically always 2 weeks shorter than gestational age. It's a really important distinction.
So using embryonic age, let's track these incredible milestones of human embryogenesis. On day 1, as we just saw, fertilization forms the zygote. By day 3, the cleavage stage is well underway. This means the cells are undergoing rapid mitotic division. They're multiplying like crazy, even though the organism itself isn't really growing in overall size yet. By day 6, it is transformed into this hollow structure called a blastocyst, which literally hatches from its protein shell and implants itself securely into the endometrial lining of the mother's uterus. Fast forward to week 9 of embryonic age, which, remember, doctors would call week 11 of pregnancy, and the embryo is about 18 millimeters long. All the essential organs have begun to form, hair follicles are popping up, and amazingly, you can actually detect a fetal heart tone using a Doppler monitor. It's truly wild how fast this all happens. Which brings us to our final part, Section 5, Assisted Reproductive Technology, or ART.
Sometimes, nature's highly specific timeline faces physiological roadblocks. And when the natural process struggles, modern medical science steps in to assist. The most well-known of these technologies is IVF, or in vitro fertilization. This procedure essentially takes that fertilization event we just watched happen inside the fallopian tube's ampulla and safely replicates the whole thing outside the body. Doctors use ovarian stimulation drugs to mature multiple eggs at once, retrieve them, fertilize them with sperm in a lab, and then carefully transfer the resulting dividing embryos directly into the uterus.
So, the crucial point here is that science literally allows us to manipulate time. In an egg or embryo banking cycle, patients can go through the retrieval process but then choose to freeze or cryopreserve the eggs or embryos. This lets patients pause their biological timeline for fertility preservation, saving those gametes for months or even years down the road.
And honestly, why are these advanced ART treatments so incredibly vital? Well, because there are numerous diagnostic reasons why conception might not happen naturally. We're talking tubal factor infertility, where blocked fallopian tubes physically stop the sperm and egg from meeting. There's male factor infertility, which involves low sperm count or motility issues. Ovulatory dysfunction, where the ovaries just aren't releasing an egg regularly. Endometriosis, a painful condition where uterine-like tissue grows outside the uterus. And sometimes it's just unexplained infertility, where doctors can't find a definitive cause in either partner. ART provides a brilliant scientific pathway right around these biological hurdles.
So, take a step back and think about it. From the continuous daily production of millions of sperm to the highly regulated release of a single monthly ovum, from microscopic cellular divisions happening in days to the modern miracles of in vitro fertilization, isn't the journey of human conception truly one of the most remarkable processes in all of biology? It is this perfectly balanced, delicate sequence of events that we are so incredibly lucky to understand and, when needed, to assist.
I really hope this explainer helped demystify the biology of reproduction for you today. If you found this breakdown helpful and want to keep exploring the science of life with us, please like, share, and subscribe to BIOCORE Notes. We have plenty more deep dives into the fascinating world of biology coming your way. Keep learning, stay curious, and I'll see you in the next explainer.